In this video, I'll walk you through the process of inverse PCR. Inverse PCR is a method used for detecting the location of insertion of a transposable element in genomic DNA. The goal of this procedure will be to amplify the end of the P element and its adjacent genomic sequence, which we can then sequence using a sequencing reaction. So our starting point looks like this. Our transposable element inserted into an unknown area of the DNA. We can see the 5' and the 3' ends of the P element here and the surrounding genomic DNA. And we want to amplify and sequence these two areas so we can compare them to the genomic sequence stored on flybase and figure out where the P element is in our fly. We will start by using DNA extraction to isolate the DNA from the cells of the fly. After that, we will complete a light restriction digest of the genome using a restriction enzyme that cuts both within the P element and relatively frequently within the genome. When that happens, we get pieces that look like this. You'll notice that we use a restriction enzyme that cuts, leaving sticky ends. These ends hanging over will be important in the next step. So once we have our pieces, we complete a ligation reaction, allowing the enzyme ligase to bring the sticky ends together. The goal is for this piece and this piece to each self-ligate, creating circular DNA. Okay, so let's take a closer look at this piece of circular DNA. What do we have? We can see the 5' end of the P element and then its adjacent genomic sequence, and as we follow that genomic sequence away from the end of the P element, we eventually hit the restriction site, which leads you back to the middle section of the P element DNA. Okay, now we've set ourselves up to complete PCR. All we need is 2 primers that anneal within the P element and extend in opposite directions toward the genomic DNA. So we have a primer that anneals toward the end of the P element and extends this way, and the other primer anneals within the central portion of the P element and extends in the other direction. As the reaction proceeds and cycles through a typical PCR, you end up with many copies of this piece of linear DNA. Let's make sure we know exactly what we're looking at. This segment is the 5' end of the P element, which then flows into the adjacent genomic sequence. This is the restriction site, where the genomic sequence ligated with the central part of the P element. So once you hit the restriction site, you're back in P element DNA. It's important to note that you can complete inverse PCR to amplify the sequence adjacent to either end of the P element. In the example I showed you, we focused on the 5' end of the P element. If you want to complete inverse PCR of the 3' end of the P element, you would just use a different primer pair that is specific to the 3' end of the P element during the PCR step of the process. So inverse PCR ends with a product of many copies of this piece of DNA, which you can then move forward with using a sequencing reaction to identify where this portion of DNA is in the genome. In the next video, I'll walk you through how that sequencing reaction works.